Deoxyribonucleic acid, or DNA for short, is essential in all living things. Every living thing has DNA, even though everything has the same thing named DNA, but DNA in each and everything is different from one another. In fact, this DNA is what differentiates one living thing with other living things. In a way, DNA can be regarded as a recipe book for making protein. DNA by itself cannot do anything to our body, but everything happening inside our body was in fact the work of protein produced by the recipe from DNA. That's why DNA is important. And DNA has the iconic double helix structure composed of 4 types of building blocks: adenine, thymine, cytosine, and guanine. And these building blocks are held by the sugar phosphate backbone, resembling the shape of a ladder. And due to the angle of the attachment, the shape becomes helix, as we all know. DNA can be extracted from living things, and in fact, scientists nowadays are so advanced, they have been tempering and modifying the DNA of living things around us, especially the microbes and the bacteria, to benefit us humans. And in this home-based experiment video, we will be extracting the DNA from a banana so that we could observe the DNA itself.
Firstly, we need smashes banana, soap to wash dishes as a detergent, salt, and alcohol. So I don't get the pure alcohol, but this sanitizer have sanitizer have 70% of isopropyl alcohol. Therefore, it is quite good to use as the alcohol. The first step is we need to smash the banana. So I have already smashed it. So we go to the next step. Step is we mix the detergent with the salt.
Today I will talk about our experimental result. Before that, let me show you about our result of our experiment. This is our result once again from Ari's work. You can see the changes. The results show that cloudy substance appear on the surface of the solution. The cloudy substance represents the DNA string of the banana. Maybe this picture is not clear enough. Let me show you the clear one. You can see the DNA strand or DNA string that been isolated from the banana. Why is this happening? First of all, why do we need to crush the fruit? Simply because the banana or every plant has a cell wall. By crushing the fruit, we actually crushing or break the cell wall to get the inside the cell wall. That means the membrane, the nucleus, or inside or gas. What does this detergent do? This detergent disrupt the cell and nuclear membranes of each cell to release the DNA. It does this by dissolving lipids and protein that hold the membrane together. As you can see in the picture here, you can see where the nuclear membrane located at. At here, this detergent will simply attack the nuclear membrane to get the DNA inside the nuclear membrane. Next is, what is the function of the salt? The salt neutralizes the negative charges on the DNA and thus enables the DNA strands to stick together. It also causes proteins and carbohydrates to precipitate. And lastly, what is the function of hand sanitizer? The other information that can be given here is hand sanitizer contains most of the isopropyl alcohol. Yes, in hand sanitizer there is a 70% isopropyl alcohol. This isopropyl alcohol is very important. This is because everything except the DNA will dissolve in alcohol. Alcohol puts water from the DNA molecule so that it then collapses on itself and precipitate. Then the DNA will become visible as white mucus strand.
As for conclusion, in this experiment we've been trying to extract the DNA from a banana. Some procedure had been conducted, like smashing the banana, mix the crushed banana with the DNA extraction liquid, and adding a layer of alcohol on top. Finally, we could observe the DNA emerge from the mixture. The DNA coagulation are not that clear in the alcohol, and we expect it is because the alcohol we use are not that pure. But anyway, we consider this experiment is a success regardless the unclarity, and we still managed to observe the DNA up close. That's all from us. Thank you.